zebrafish embryo acute toxicity testing to predict short-term toxicity to fish and beyond, which is the 6th webinar in our series on alternative approaches, co-sponsored by the PETA International Science Consortium and Chemical Watch. I'm Gilly Stoddart of the PETA International Science Consortium, and I'll be moderating this event. Today we have 3 speakers who will present for about 45 minutes before we go into a 15-minute Q&A session. Delegates can ask questions during the webinar by using the question box on your screens. Send your questions in as early as possible, as I can then forward them on to the speakers in good time. If your question is not answered during the webinar, please submit it into Chemical Watch's forum, and we'll address it there. We'll provide the link to the forum at the end of the webinar. If you experience any technical problems during the webinar, you can also use the question box to send a query to Lorna from Chemical Watch, who will be able to assist you. Delegates should also note that a recording of this webinar, along with the slides, will be available on the Chemical Watch and the PETA International Science Consortium websites in the next few days. I'll now give you a little bit of background to this webinar series. The goal of this 6-part webinar series is to help registrants plan for the 2018 REACH deadline. Hundreds of thousands of animals have already been used for REACH testing, and millions more are expected to be used in the coming years. However, there are alternative methods that can be used to meet REACH data requirements while minimizing animal testing. For more information on minimizing animal testing for REACH and recordings of the past webinars, please feel free to visit the PETA International Science Consortium website. I'd also like to let you know that our scientists are available to assist companies in minimizing testing on animals, so please get in touch with any questions after today's webinar. I'd also like to let you know about a bonus additional webinar in this series that will be held in June. During this webinar, Derek Knight from ECHA and Karen Killian from the European Commission will talk about their perspectives on the use of non-animal methods to meet REACH data requirements. Today our speakers are Drs. Marlies Halder and Scott Belanger, and also Professor Thomas Brownback. Dr. Halder is a senior scientist at the European Union Reference Laboratory for Alternatives to Animal Testing of the European Commission's Joint Research Centre. She is responsible for EURL-ECVAM's activities related to the quality control of biologicals and environmental toxicology. EURL-ECVAM coordinated, with the support of a validation management group, the OECD validation study on the zebrafish embryo acute toxicity test. Dr. Belanger is a research fellow at Procter & Gamble's Corporate Global Product Stewardship Safety Organization, where he directs research on ecological and toxicological responses of fish, invertebrates and algae for consumer product chemicals. Scott has published widely on the correlation between the acute fish toxicity test and the fish embryo toxicity test. Professor Brownback is the head of the Aquatic Ecology and Toxicology Center at the University of Heidelberg. In recent years, Professor Brownback has focused on the use of fish embryos as alternatives to adult fish, and his laboratory played a pivotal role in the validation of the fish embryo toxicity test. In this webinar, Dr. Halder will begin by describing the REACH data requirements for short-term toxicity testing on fish and the OECD acute fish toxicity test. She will then describe the fish embryo acute toxicity test, which was adopted by the OECD in 2013 and is recommended by ECVAM as a possible alternative to acute fish toxicity testing. Dr. Belanger will provide an assessment of the correlation between the OECD acute fish toxicity test and the fish embryo toxicity test. This will include an assessment of the domain of applicability and the possibility of using the fish embryo test in traditional fish quantitative structure-activity relationships. Professor Brownback will then describe the potential use of fish embryos beyond acute fish toxicity testing and provide a view to potential future uses of fish embryos in a much wider array of environmental science and human health applications. With that, I'll hand over the controls to Marlies to start.
Thank you, Kelly. Good afternoon, everybody. First, I would like to thank PETA and Chemical Watch for arranging this webinar and for giving us the opportunity to speak about the use of fish embryos. Next slide, please. As you know, REACH requirements are tonnage-driven. For the aquatic toxicity, the various REACH annexes list the following standard requirements. short-term toxicity testing on invertebrates and growth inhibition on aquatic plants in Annex 7. Our endpoint of interest, short-term toxicity testing on fish in Annex 8, and long-term toxicity testing on fish and invertebrates in Annex 9. I would like to underline here that acute fish toxicity testing may also be required by other pieces of legislation, for example for biocides, plant protection products, pharmaceuticals, feed and others. Next slide, please. According to the recent ECHA report on the use of alternatives to testing on animals for the REACH regulation, 3,662 substances are registered for tonnage levels greater than 100 per year. With regard to short-term toxicity to fish, experimental studies were available for 62 of the cases. Alternative options used the rate of evidence with 10.5%, this is the light blue bar, read across with 21.7%, the dark blue bar, QSAS with 2% in dark gray, and the study was omitted in 3.7% of the cases, indicated by the light gray bar. From the data submitted, ECHA concludes that 645 new acute fish toxicity studies had been conducted since 2009. Noteworthy, registrants are not required to submit testing proposals to ECHA for short-term fish toxicity studies. The final REACH deadline is in 2018, covering substances from 1 to 100 tons production or import per year. According to ECHA, 25 to 50,000 substance registrations are expected. Next slide, please. The current standard method for short-term toxicity to fish is described in the OECD test guideline 203, Fish Acute Toxicity Test. The test can be carried out with a range of fish species: zebrafish, fathead minnow, Japanese medaka, rainbow trout, bluegill sunfish, common carp, guppy, and others. Juvenile or adult fish are exposed to 96 hours to at least 5 concentrations of a chemical, and the concentration lethal to 50% of the fish is determined. Water and, if needed, solvent controls are run in parallel, and 7 to 10 fish are used for treatment. It is, however, possible to conduct a limit test at the concentration of 100 mg per liter when fish toxicity is not expected to occur. Next slide, please. Now let's start with our possible alternative to the acute fish test, the fish embryo acute toxicity test. It took almost 10 years from its initial inclusion on the OECD test guideline work plan in 2004 to the final adoption of the test guideline 236 in 2013. During this period, a validation study was carried out to assess the reproducibility within and between laboratories of the ZFET, and 20 chemicals had been tested and 11 laboratories participated in this exercise. The 3 speakers, as already mentioned by Billy in this webinar, had been involved in the validation study and test guideline development. Next slide, please. This slide gives you a schematic overview on the ZFET. Starting from the left with the production of eggs, their collection and immediate exposure to the respective test concentration, selection of fertilized eggs, and distribution into 24 well plates. Several aspects of this test guideline are comparable to the fish acute toxicity test. At least 5 test concentrations are used, and the concentration lethal to 50% of the embryos is determined. Moreover, positive, negative, and solvent control, if needed, have to be run in parallel. The use of at least 20 embryos per concentration and control is recommended. The embryos are exposed to 96 hours, which means that this is well beyond hatch. During the course of the test, lethal effects should be recorded at 24, 48, 72, and 96 hours. 4 observations indicate acute lethality, which are coagulation of the embryo, lack of somites, lack of tail bud detachment, and lack of heartbeat. Next slide, please. As shown here, a live zebrafish embryo is completely transparent, and any disturbance of the development can be observed using light microscopy. In the upper row, you see a normal developing zebrafish embryo, and below, 3 of the 4 possible observations indicating death of the embryo. Coagulation may occur within a few hours after start of exposure or throughout the exposure period and indicates general acute toxic effects. Somites should be visible from 10 hours post-fertilization onwards. In their absence, the embryo will not further develop and die. The tail should be detached from the yolk at 24 hours post-fertilization, indicating normal growth of the embryo. The 4th observation, not depicted here, is lack of heartbeat. The heartbeat is easily detectable from 20 hours post-fertilization onwards, and its absence indicates death of the embryo. Next slide, please. There have been a lot of discussions on how the use of a zebrafish embryo of zebrafish embryos would be considered under animal welfare legislation. When we drafted the Electron recommendation on the ZFET, we consulted our colleagues at DG Environment, being responsible for Directive 210/63 on the protection of animals used for scientific purposes. The 2 boxes on the right side of the slide display the relevant information. Which animals are under the scope of a directive? These are live non-human vertebrate animals, including independently feeding larval forms, and that a zebrafish should be counted 5 days post-fertilization. The second statement is taken from the Commission implementation decision on a common reporting format on the animals used in Europe. Considering this, and that the OECD test guideline 236 and that in OECD test guideline 236 zebrafish embryos are used until 96 hours post-fertilization, the embryos in question should not be considered as independently feeding larval forms within the meaning of a directive, and therefore they do not fall within the scope of a directive. Next slide, please. I have already mentioned the Electron recommendation on the ZFET published last year after consultation with our stakeholders and the public. We concluded the following. The ZFET is reproducible. It provides information on acute fish toxicity comparable to the information derived with a standard method. The ZFET is ready for regulatory use as alternative to the fish test, and we further recommended the update of relevant guidance and legislation. Since there has been a lot of discussion on the relevance of the potential limitations listed in the OECD test guideline, it may be useful to draft an OECD guidance document when more experience is gained with the method. Such a guidance document may address, apart from technical aspects, the following: the role of the metabolic capacity or profile of fish embryos in comparison to juvenile or adult fish, the decreased bioavailability of high molecular weight chemicals not passing the chorion. There is concern that the toxicity of such chemicals could be underestimated since the exposure time is reduced to the post-hatch period. At the moment, OECD test guideline 236 recommends to not use the fish embryo toxicity test in these circumstances. For chemicals with specific mode of action, additional sublethal observations may be included and improve the prediction of the fish embryo toxicity test. In the following, Scott Ballinger will report on the predictive capacity of the fish embryo acute toxicity test and will refer to the fish FED database. This database is a valuable source of information. It should be maintained and updated. This would, in my opinion, support regulatory acceptance of the method. With this, I would like to hand over to Scott Ballinger.
Thank you, Marlene. Before I begin this section, I would also like to thank you all for joining us today, listening in to this work on fish embryo testing and what this can mean for both research, for chemical hazard assessment, and of course its impact on animal welfare. Next slide. So the first thing that we are faced with here is really, does the fish embryo test predict acute fish toxicity? And in short, it really doesn't do us any good if we develop an assay that is not predictive, even if it operates perfectly well. So that's really what the focus of this part of the investigation became. What does success look like? It looks like perfect success when the FET predicts fish acute toxicity 1-to-1 for the same chemical. It would be predictive for all chemicals equally well with better statistical power. The chemical coverage is very, very wide, and that we can repeat those results time and again. Now, aspects of this we gain through the validation, but the questions we have asked here are a little more generic. And next slide, please. Environmental toxicology has a very long history of improving testing through shortening of tests, developing additional endpoints that we can use to understand environmental hazard, and do this in a way that provides insight that is at least equivalent, if not improved, over time. As an example, back in the late 1960s, fish full life cycle tests were the test of choice for evaluating chemical toxicity of fish. This eventually became shortened to what we now know as the OECD 210, the 30-day or 90-day growth study with fish like fathead minnows and rainbow trout. The fish embryo test is just the latest series in this long list of advancements. And this is a product of over 20 years of research wherein we generated a lot of information. And we can take advantage of this to improve animal welfare and the science as well. Next slide. So what we do is we ask how much information is out there. To make the transition from traditional fish toxicity testing to fish embryo testing, we have to show that predictive nature. That requires data. Is it available? How variable is it? Do these things coincide with accepted notions of fish tolerance or sensitivity? And even things like structure activity. Do fish embryos obey structure activity the way fish do? To make that transition, we need to develop these relationships in a very well-coordinated way, and that's what we'll explore over the next several slides. Next slide. So this was the general process. We had a very large data collection effort, involved many labs, lots of historical literature, and in the end, we gained about 2,500 compounds or 2,500 studies. About 2/3 or a little over, not quite 2/3 were done with fish, and the remainder with fish embryos. Very wide chemical space, 17 modes of action, lots of chemical categories, and then we evaluated what were the qualities of the chemical domain that we were looking at. And then we evaluated fish toxicity relationships with the FET by chemical class, mode of action, test design considerations, and so forth. And in the bottom, we're asking, does the fish embryo equal the fish? Next slide. So a few words about the chemical space that we're in here. The data compilation was current circa 2012. And which even now could be further updated. We continue to gather information. About 2,500 studies, over 200 chemicals, 17, 18 modes of action, 38 chemical categories. A total of 151 chemicals had, in this assessment, have both fish and fish embryo data. And a handful of those were deemed not suitable for evaluation due to data quality. So in the end, it ended up somewhere closer to 140. But this was then the suite of studies we began to probe. Next slide. A little bit more about the chemical domain. Here we're looking at histograms of molecular weight, log KOW, and solubility across fish and fat studies. And one thing you should very quickly see is that the domain is very broad. Log KOW, for example, spans almost 11 orders of magnitude. There's a lot to go on here. I will mention that the validation program touched on the edges of both the high and low range of solubilities, molecular weights, and KOWs. And from this, we can conclude that when we do develop these relationships, these regressions, we are covering an awful lot of chemical space. So the results ought to be fairly broadly applicable. Next slide. A particular component of this analysis that was of interest to us was the relationship between fat and fish toxicity to log KOW. It's so central to environmental risk assessment, so we pay a lot of attention to it. High KOWs are generally low in solubility, high in bioaccumulation potential, and also relatively toxic. So what you see in this XYZ plot is on the floor of the figure is a far view of the fat-fish toxicity relationship. We'll look at that more in a second. But in the z-axis is log KOW. And what you can see is that there's generally a trend towards log KOW values that are low being less toxic, log KOW values being highly toxic, and that there's quite a nice spread. And we don't see any inherent bias in this kind of information. Meaning, for example, we do not see high KOW compounds that have a low order of magnitude of toxicity of fish embryos. Next slide, please. So here's the real meat of this portion of the webinar. First, a little orientation for the slide. On this graph are a whole bunch of lines. A very light blue line running, bisecting the graph diagonally is the line of one-to-one correspondence. The dark blue line is the regression of a fish toxicity to fat. The dark circles are the geometric mean of the XY coordinate for the fish and fat data on a particular compound. The light blue circles are those individual compound data in the plane that you see the data going. So what do we have here? We've got a fish LC50 having a highly significant regression compared to the fed LC50. And in fact, the slope of the relationship is 0.989. It's as close to unity as you can get. An intercept of 0.195, very close to 0. This is a near perfect relationship. The correlation coefficient is 0.95, and basically this is about as good as it can get. This particular graph is confined to 96-hour FET data and 96-hour fish data with 72 compounds embedded. And in practical terms, you see the numbers off to the right. These are really solid predictions. Next slide, please. Another way to look at this is to think about those fat fish regressions relative to fish comparisons. And we have this ability in the database to go back and look at this. In the fish portion of the database are several species: Japanese Medaka, rainbow trout, bluegill, fathead minnow, zebrafish. And this complicates matters. It makes the regressions more variable than they otherwise might be. All of these species, though, are OECD recommended. So what we did was look at the fish-to-fish relationships for all possible combinations here. And what we see is that they are just like the fish embryo-to-fish regression that you just saw in the previous slide. There are a few trends that come out. Rainbow trout, slightly more sensitive than most species. Japanese Medaka, slightly less sensitive. We can see this more quantitatively in the next slide. Next slide, please. Where here we have the frank comparisons of slopes and intercepts for binary combinations of fish or fet. And highlighted in yellow are the fet-to-fish relationships. And you can see that the fet slope and intercept coefficients look just like fish-to-fish slope and intercept coefficients. And in fact, if we put a name like fish number 6 here, you would never know the difference as opposed to calling it a fet. So indeed, the fet operates like a fish and behaves like a fish in terms of acute toxicity information. Next slide. So the next part, as we near the end of this section, is to ask the question, might QSARs work? Also, a very important consideration from a technology guidance point of view, looking at new chemicals or looking at things that we know very little about. Next slide. So here, what we did was we went back and probed some standard fish QSARs. And in black, we have the US EPA EcoSAR QSAR for neutral organics plotted. And on top of it, in red, is the regression for neutral organic compounds found in the fish portion of the FET-FISH database. And what you see is that these are virtually identical. In other words, the fish data in our database behaves exactly like EcoSAR would predict. Okay? Next slide. We layer on top of that the fish embryo test data. And here, again, we get very similar results. These are the ones in blue. The slopes are not significantly different. The confidence intervals for all 3 lines envelop the regressions for the other components in this work. And voila, we have fish QSARs that we can apply to fish embryo test data. Now this is one example, but it works for other classes of compounds as well. Next slide. So where are we in this analysis? The available data that we have is extremely rich. The most commonly tested species we have in our database are those that are prominent in OECD and reflect geographic preferences. Canada, the UK love rainbow trout. The US loves fathead minnow. Europe uses danio, zebrafish, and Asia often uses the Medaka. We've got a very large database, and we've got a large range of large span in the chemical space that we're evaluating here. Next slide. The sources of variability that could contribute to noise in this relationship are enormous. Fish and FET, though, are subject to the exact same influences, and the FET data is actually more homogeneous because danio is the most tested species. But this is changing. More other species are being used now. QSARs work. We don't have a full analysis of this yet, but it will happen. We are actively using this model in industry chemical hazard assessment. Decision making is being done using this, and we're very confident in the assay and aware of the animal welfare commitment and considerations it represents. I can also say that most major contract labs in the U.S. and in Europe are already offering the assay following the adoption of the 236. So that concludes this part of the seminar, and I'll hand it over to Thomas, Dr. Brombeck, who's going to talk about other applications of fish embryo testing.
Yeah, hello, and thanks for listening from my side as well. The next slide, please. So far we've talked about the use of the fish embryo test as a surrogate to OECD test guideline 203, but we should bear in mind that similar test scenarios are being used as well. For instance, range finding tests for higher tier testing, prolonged toxicity, OECD 210 to 12, also for endocrine disruption 229 to 30 to 34. And the same holds true for the future OECD test guidelines like the MEDACA extended one generation reproduction test. For all of these, the fish embryo test can equally replace TG203. So there are quite a few more applications for the fact. But apart from these, as a person working at a university, we are predominantly interested in, well, looking at other possibilities to use the fish embryo test. Next slide. And I will give a few, well, selected examples where we can use the fish embryo test in a modified version of the protocol of the fish, of the FET 234, 36, sorry, for instance, heterogeneity testing, neurotoxicity testing. We can look into biotransformation, we can look into endocrine disruption, we can scan for genotoxicity, and we can certainly do what many colleagues do, we'll look at gene activation. Next slide. If we start with heterogeneity, we have a major advantage already mentioned by Marlies Halder, and that's the fact that the zebrafish embryo is perfectly translucent. And we can stain for bones in red and cartilage in blue, and we see a nice picture of the developing skull in a zebrafish. Next slide. Now, if we expose the zebrafish embryos up to an age of 120 hours, so within the scope of animal welfare legislations in Europe, we can see, as this example shows with dithiocarbamates and hydroxides, a progressive degeneration with increasing concentrations of these known teratogens. The red, the bones really disappear, and you see nicely the deformation of the cartilage. So embryos as methods for heterogeneity testing, not only for fish, but potentially also for mammals. Next slide. Another issue which is increasingly being discussed in aquatic toxicity testing is neurotoxicity. On that slide you see a nice picture of the head of a 96 hours old zebrafish with the neuromasts highlighted. The neuromasts form the lateral line system of the fish. That is a sensory system for the detection of potential enemies or even prey. Next slide. If we now expose these embryos to certain known neurotoxins like copper sulfate, neomycin, or peroxon, we can easily detect a degeneration of the neuromasts, which means at least partial degeneration of a major sensory system of these embryos. Next slide. And on this side you can see that, again, we get a fairly neat dose-response relationship between the agent, in this case copper sulfate, and the reaction with 10 micromolar, which is fairly low, copper sulfate. it is almost impossible to detect any neuromast. So they have lost their lateral line sense. The next example, next slide, is on cytochrome P450 induction, which is a major issue that has been discussed extensively when it came to the applicability of the fish embryo test, since there are substances that need to be bioactivated by cytochrome P450. So what we did is we designed a test showing in living embryos the induction of P450, in this case by a standard inducer of P450 beta-naphthoflavone. And it's a 32 hours old embryo. And you see at the orange arrow, the eye, at the red ones, the red arrows, the circulation, and the Ihranlage at the yellow arrow. So P450, in this case, P452B is clearly there. Next slide. What about earlier stages? This is a 8-hour-old embryo, and again, induction by beta-naphthoflavone of cytochrome P452B, and it's quite evident that in surface view and in transverse section, there is quite a lot of cytochrome P450. Next slide here. In fact, even if we look at the controls, that is, without induction by beta-naphthoflavone, we also see a signal. And if we go back to literature, we can learn, it's a 1979 paper, that cytochrome P452B is actually a controlling agent in development. So it's expressed constitutively. So P452B is there. Next slide. What about P451A, the major biotransformation enzyme in fish for many environmental contaminants? This is an example of a 72 hours old embryo which has been exposed to sediment extracts from the Rhine River. The EC10 for externally visible changes is 4 milligram SEQ, sediment equivalents. And you can see that even quite clearly below the EC10, there is a clear signal of, in this case, the liver, which is actually being activated at around 72 hours. So the liver, as soon as it emerges, is active in biotransformation. P451A is there. Next slide. However, a little bit surprising, if we expose these 72 hours old embryos for 3 hours only, we get a much stronger result. Actually, I think this is an important finding, since this shows that the system is easily overloaded. So many studies that failed to detect P450 in the embryos may have failed because they had applied much too much high concentrations. The next slide, please. This is endocrine disruption. It's been published in 2003, and it shows which proteins can be visualized in the blood in zebrafish embryos. Well, estradiol receptor alpha is important for development, so we can find it as early as 1 day after fertilization. Pretty young embryo. Estradiol receptor beta shows up 9 days. So an apellucida protein, which is a structure protein of the eggshells, appears after 22 days. And vitellogenin, that's the fourth lane, an important biomarker in endocrine disruptor research, it can be visualized as early as 4 days after fertilization if we expose the embryo to ethanol estradiol. So again, the embryo is a surrogate for, well, adult fish. Next slide. Endocrine disruption is not only estrogens, it's also, for instance, thyroid disruption. So we talk about goitrogens. This is a picture of a little bit older embryo, and you see at these blue arrows The pinkish ovals, that's the thyroid follicles. Next slide. If we expose to a known goitrogen, you see a tremendous proliferation in the number of follicles and in the volume of the follicles. We're just about to compare this reaction to what we see, for instance, in the Xenopus assay. And it looks as if the zebrafish embryos, even at an age of 120 hours post-fertilization, are just as sensitive as are Xenopus larvae. Next slide, please. Genotoxicity testing, at least in Europe, does quite a lot of genotoxicity screening in sediments in the environment, and we tried to use our zebrafish embryos. For the comet assay, for instance, we need single-cell suspensions, and it is quite easy to get such suspensions just by mechanical disruption of the embryos. And as you can see in this example, you get again a fairly neat dose-response relationship between the concentration of the sediment extract and the tail moment, that is, in the positive result in the comet assay. Genotoxicity testing. Next slide is a very common approach. For instance, microarrays. We talk about the induction or the repression of certain genes, and again we use embryos. I'm not talking about the technique itself. It will be known to many of you. Next slide shows that if compared to 12 model compounds, the lower two lanes show by far the highest number of repressions or inductions of certain genes. These are again Rhine River sediments. So again, for microarrays or for PCR methods, induction of certain enzymes can easily be visualized. And when we talk about new trends in ecotox, these gene changes will certainly receive more interest than in the past. So this was just a very brief rush for a series of examples how we can use these embryos beyond acute toxicity testing. The next few slides are just a listing of important references. Thank you from my side.
Many thanks. And over to you, Philip, to start the Q&A session. Thank you.
Okay, well, thank you very much for all of our speakers. I'm sorry I wasn't able to join you earlier. Perhaps we can begin with some very good questions here. Perhaps I can ask Marlies Holder to answer this question about when this test will be integrated into the REACH regulation.
Thank you for that very important question, and I must say that I don't know when it will be integrated in the REACH regulation. I can't tell. However, there are some signs coming from ECHA that the use of this method will be possible in the future. So ECHA is carrying out analysis of the relevance and adequateness of fish embryo toxicity tests to fulfill the information requirements and address concerns under REACH. You can find this information on the ECHA website. At the same time, I think it was published in Chemical Watch on the 13th of February this year, that the member state, ECHA member state committee, actually points out to a registrant that there were actually requests to carry out for a given substance an OECD 203 test, and at the same time they underline that there is an an alternative method available now with the fish embryo toxicity test. However, they also underline that it's up to the registrant to make a case for using the alternative. Maybe Thomas and Scott want to add something?
Or perhaps there's somebody from ECHA listening who might like to add something. They can make themselves known to the organiser. I'm sure we can listen to them.
Yeah, this is Scott. Am I on?
You are.
I would point out that the use of fish embryo tests was foreseen in the REACH guidance documents. Of course, this is not quite the same thing as fully accepted, but it clearly was part of the discussion as far back as 2008. And it was equally well placed in the OECD fish testing framework, whereas Thomas talked through, there are many opportunities where it has a positive impact on the numbers of fish used, in addition to a standard 236 format, but as a component of preliminary work for fish chronic tests, for example. So I think the idea that it will require the registrant to make the case, I think the information is there to do that in general, and we look forward to seeing some positive results from those applications.
Okay, well that is obviously a key question for ECHA to answer. They maybe don't have that answer at the moment, but the sooner that we have it officially integrated, the better. I'm sure that Chemical Watch will tell you when that happens. Perhaps we could move on to another question on question of which species are to use. I mean, this question may have been asked before Thomas Brownback completely explained it, but perhaps he would like to consider, the question is, what are the options on using other species such as fathead minnow instead of zebrafish? Responses shown to be are shown to be the same, and some labs have trouble culturing some species such as zebrafish.
Well, I can say that there's a growing body of work on fathead minnow particularly. Probably in the neighborhood of, at this point, close to 100 studies that I'm aware of. And the developers of the 236, of course, had to grapple with this concept also. And the 236 is very directly a discussion of zebrafish, with the caveat that other species will require some modification to make them fully utilizable in the same context. And I think we are seeing great evidence of that being successful for fathead minnow. Japanese Medaka and rainbow trout are going to be a different story. Their developmental times are markedly different. The size of the eggs are larger, and they take longer to develop. And of course, bluegill are rarely cultured in the lab, so they're almost off the table in terms of similar discussion. But I think fathead minnow, I think we're seeing great progress there.
Right, this is Thomas Browneck. We published the first results of comparisons between the fat amino, the zebrafish, and the Medaka in 2009. And in the meantime, we tested, especially with fat amino, about, well, 24 substances. And for, I would say, well, normal model compounds, the results are fairly comparable if we adapt the protocol. For the fat amino, adaptation of the protocol is fairly easy, and as Scott pointed out, the protocol for the Medaka is well possible. It needs some, well, amendments when it comes to the time schedule. But basically, the fat protocol can be applied to fat amino and Medaka just as it can be applied to the zebrafish.
Okay. Well, there's clearly developing options there, and no doubt this will continue. Thank you, Thomas. Moving on, another question for Thomas, I think, was how predictive is the fish embryo toxicology test to chronic toxicity?
Well, given the fact that there is an almost perfect correlation between the fat and the fish test, the predictive power of the fat is just as good as it is the predictive power of the fish test. Actually, we've never analyzed that. Since we extrapolate from acute toxicity to chronic toxicity, which is highly questionable, by the way, and if fat equals fish, the predictive power should be comparable. But again, I would doubt whether it makes sense to predict acute, to predict chronic toxicity from acute toxicity data, be it fish or fat.
Okay. Anyone else?
The flip side of that is it is a regulatory construct that we do that, and there certainly are many analyses in the literature where a fish acute toxicity is related to chronic toxicity through acute to chronic ratios of various levels of sophistication. It isn't as simple as the typical regulatory construct might have one believe, but it is done. And I think the point from Thomas's comment is that if you feel comfortable doing that with acute toxicity to fish, you should feel equally comfortable or uncomfortable doing it with fish embryo toxicity.
Thank you, Scott. Another question. Well, recognize the endocrine disruption effect. Hormonal disruption would be combined with apical observation. How is PET adapted to get the apical effects? Thomas, would you?
Yes. Well, that's certainly hard and difficult to answer. In the embryo, there are no further apical endpoints. Well, endpoints typical of endocrine disruption. It's just for the biomarker answer, that is the vitellogenin induction. So we do not see differences in sexual differentiation at that age. We need to wait. In zebrafish, the gonad differentiation should be finalized at latest 60 days after fertilization, which is certainly beyond the scope of a protocol like that of the fish embryo test. It's just for screening purposes, for looking at certain endpoints that indicate that the compound in question might be an endocrine disruptor.
Thank you. Yes, it's an interesting observation. I have a question here for Scott. Where complex mixtures of chemicals are included in the pet or fish relationship study, were complex mixtures of chemicals included in the pet-fish relationship study which you discussed, Scott?
No, really, that's a very, very logistical thing to think about, because imagine it would have to be a tacit study of a fish embryo test done side by side with a standard fish acute, with the idea that you were probing that complex mixture question. So in terms of a literature review, this kind of opportunity is extremely rare. That said, there is a great deal of work that has been done on use of fish embryos to understand effluent toxicity. In fact, there's a German DIN on and an ISO standard guideline addressing that. So it's not that we cannot. It has been done with complex mixtures, but to do the direct comparison is not really something we've seen. Particularly here in the U.S., acute fish testing per se for effluents and complex mixtures is not a very common practice. It's much more of a chronic exposure and chronic toxicity type of question. So in countries with more advanced treatment, rarely do we see acute testing done for that purpose.
Thank you, Scott. One can see how that works. Moving on to, we have another question here for the a question here for Thomas. Is the mode of action to kill adult fish and embryos the same with the same test chemical? So, searching question.
Well, that's again hard to say. I would assume this depends on the mode of action. But normally, as far as we do have data available, it seems as if the modes of actions are fairly similar. It is certainly right if you look for a mode of action which only hits the adult fish during, for instance, sexual reproduction, it would not be seen in the fish embryo test. But this is not an issue of acute toxicity. So with respect to 203, we do have evidence that it's the same mode of action, but there might be exceptions.
Yes, indeed. Yeah. Another question, Thomas. Are the embryos I'm not sure I can understand this word, chorionated, is it? Chorionated?
Yeah. We do have a protocol for dechorionation of the zebrafish embryo, of the fathead minnow embryo, but in the routine procedure we do not dechorionate, just for reasons of simplicity. And for normal compounds, say with a molecular weight lower than 3,000 or say 2,800, usually decorionation is not necessary at all. But if there is evidence, for instance, that the substance in question is charged, especially negatively charged, it might make sense to decorionate, because there are pores in the chorion, and it's like a cylinder, and the narrowest position there is a positive charge, and it could be that the test compound might be trapped right at that location. So overall, no decorionation needed for, say, standard compounds. If we talk about polymers, for instance, decorionation does make sense, and it's been published in 2011.
Okay.
But I think we should underline here, as Thomas already said at the beginning, that the OECD test guideline 236 does not include the decorionation. So if you want to run this would be a deviation from a test guideline, and if you want to use it for regulatory purposes, it might not be, the results might not be accepted. So you have to be very careful in doing this.
Actually, during the design of TG236, particular importance was given to the fact that with an observation time of 96 hours, we usually cover hatching of the embryo. And we do have examples where we see that the test substance does not show an effect before hatching, and as soon as the embryo hatches at around 72 hours, toxicity starts. Even though toxicity starts late, the data fit into the correlation between the fish embryo test and the fish test. So a sort of natural decorionation does occur according to the standard protocol. And as far as we know, this is sufficient to cover also the effects of high molecular weight substances, for instance.
Okay. Good. Is that no more to add on that one? We'll move on to a key question. Can you give an estimation of the cost of the test and compare it to the OECD 203? Scott, have you got anything to say on that one?
I can address that in some general terms. Of course, this is we see this occur with new assays all the time. When they're initially deployed, they're targeted at a certain cost, and that's based on the volume of business that contract research organization might require. As volume and increases and comfort with the work increases, costs do tend to change. That said, initially, I would say that the cost in general, not always, but in general, is somewhat higher than an OECD 203. Of course, with the added benefit that you're not using protected organisms from an animal welfare point of view. A wild card in this always is the analytical effort that you have to place into the test. Confirmation of substance exposure is generally required under most test guidelines if a method is available. And what we see is that the analytical methods that have been typically developed for low concentration aqueous exposure that are standardly done in a 203 are immediately adaptable to small test volumes that are used in the 236. So in the end, the analytical costs are less or are about the same. And of course, space is less. So we're in a period of a lot of dynamic cost structuring of this presently. But I think those are some general observations that we've made. Others may have different experiences as we move forward.
As Scott pointed out, it's Thomas again, in Germany we do have experience with effluent testing with a fairly similar protocol, acute testing on effluents. And we've got 10 years of experience, and as far as I know from contract labs, the costs for the test itself, that is without chemical analysis, which is not being run for effluents, the costs have gone down to about 70–75% of the costs for an acute fish test. So once adapted, once established in the labs, it's likely to be less cost extensive than the fish test.
Thank you, Scott. Thank you, Thomas. That is very interesting because we've got every possibility of a reduction in costs as we move towards the 2018 deadline. Can we move on to the next question, which is directed at Marlies. Perhaps, is there any research or studies to apply the AOP on the ZFET or vice versa?
There is a research program under the static RI which is actually looking at AOPs and chronic fish toxicity, and there fish embryos are used. But for the AOP, probably the chronic fish toxicity tests are more important than the acute fish toxicity. And I also know that it's not only this static RI program, but there are several groups are working on AOPs, and a lot of them are actually using fish embryos. So maybe Thomas and Scott, who are more in the research area than I, can add to this.
There are a couple of stabs that have been taken at this. The idea of using fish embryos for AOP development specifically, I think, is probably overall a misdirection because it's an acute study. AOPs really receive their full utility in long-term exposure, where the adverse outcome that people are interested in is survival of the population and continued development of the species, which an acute test will never do for you. However, fish embryo tests have been envisioned as a means to help prioritize what chemicals might be best used for, go into AOP kinds of analyses. And there are a number of publications on this already that have taken that tack. So I think, again, this is one of those acute versus chronic toxicity ideas that we have to really keep straight.
Okay. Time's moving on, so there are another three questions or so. One addressed towards Scott. How do you deal with poorly soluble test substances and potential absorption to well plate substrates? do you normally use static renewal for this, and what are other strategies available?
Yeah, one sees this in the data at large when you compile it. Compounds that have characteristics of being difficult test substances, not only solubility but volatility, sorptivity. The idea that people very quickly gravitate to is one of two approaches, either flow-through or static renewal. And in the 236 guideline, it's very clearly recommended that for more difficult substances, static renewal is the recommended approach. Flow-through will always be a fairly niche idea for fish embryo testing because of the complexity of the logistics of doing that. But what we do is pre-sorb the test chambers, the 24 well plates with the chemical of choice, usually for at least 24 hours, and then do 24-hour renewals, which is actually very standard in most people's laboratories at this point. And you can see this ultimately in analytical confirmation information, which, again, for difficult substances, you're almost obligated to do in order to get the exposure determination correct.
Very good. Anyone else?
Hopefully that answered the question.
Thank you. Perhaps before we wrap up, there's one more question here. How important is analytical verification of exposure concentrations? So far this has not been done in most of the fish embryo studies. Can we expect that correlation may even improve if exposure concentrations would be measured as in the acute fish toxicity test?
Well, again, in a regulatory format, it's essential and necessary to do analytical confirmation. In research, per se, you might be able to just get away with nominal exposures, although then you're always left with where is the compound really. Of course, it also depends on the exposure profile of the compound, or how close in proximity are you to a GHS classification labeling cutoff. And these situations happen all the time. So there is no real excuse fundamentally for not doing analytical confirmation of fish embryo test exposures, just like there is no reasonable justification for not doing them with acute fish tests either. And it just becomes very important under defined conditions that we take that challenge on, as difficult and as expensive as it can be. In practical terms, if you think about what you would do normally, let's say 100% of the compound dissipates in 24 hours, and you did have all the compound there at time 0. So you have a sawtooth pattern of exposure, and you take a geometric mean of 0 and your nominal or initial dose concentration. In many conditions, you will probably be off by as much as a factor of 3 in the ultimate determination of the LC50. Maybe as good as a factor of 2 if the loss is completely linear and time dependent. But you know, that gives a sense of how important it can be.
Okay. Anybody else, anything to add to that, Marlies? Or Thomas?
Yeah, well, during the development of OECD 236, we certainly looked at what we called outliers. That is, substances that did not fit the correlation between the fish embryo test and the fish test. And in many instances, it proved that it was concentration inconsistency in the system that caused these outliers. So it is absolutely indispensable to run analytics. This has been nicely confirmed in a study by the Environmental Research Center at Leipzig, Germany. They also looked at these outliers, and in any of these cases they found that the actual concentrations did deviate from the nominal concentrations. So I think at present it is state-of-the-art to run even acute toxicity tests with chemical confirmation of the concentrations, be it the fish embryo test or the fish test.
Thank you for that. Well, the time is pressing. I think it's probably time to round things up. There are a few more questions, but I think that we've covered the most important. I think I would like to wrap things up and to thank you very much, all of the speakers, and the very best of luck to all of our listeners as they go about their work in this field. And can I say, on behalf of Chemical Watch and the other organisations, PETA, involved in this, thank you very much and goodbye.